All right, so as I mentioned, we're going to move on with our next little lecture clip here, which is going to be about osmosis. Okay, osmosis is a passive process, so we're returning here to a passive process, but it's such a critical process for the function of all the cells in your body and for your survival and for the movement of fluids inside your body from one location to another that it deserves its own little lecture video clip. I cannot emphasize enough how important it is for you to try to understand now osmosis and how important it is, because it is one of the key topics that you return to over and over again during your biology classes, and you're also going to be returning to it again when you take your health sciences classes. So you want to try to learn it now. Okay, so osmosis, the diffusion of water molecules across a selectively permeable membrane. And in biology, when we're thinking about selectively permeable membranes, those of course are our cell membranes. You have water, of course, in your extracellular fluid. You have water in your intracellular fluid. And we've already seen how even though those water molecules, shouldn't like those fatty acid tails in the plasma membrane, they're small enough that they can wiggle their way across from one side to the other. And so they are able to diffuse in either direction, and that diffusion is called osmosis. All right, a couple of other terms here. Well, what in the world determines whether or not a cell is going to take up more water than it's going to lose, or whether it's going to lose more water than it takes up, or maybe those water molecules are just going to move back and forth evenly and keep everything nice and evenly distributed. So there's some additional terminology here that we need to take a look at, because as you'll see, it's really the concentrations of your solutes. That's going to be the key thing. How many dissolved substances, what's the concentration of all those dissolved substances you have in the extracellular fluid versus the intracellular fluid? That is going to determine how much water flows into or out of a cell. There's a special term for that, osmolarity. Osmolarity is a term that refers to the total concentration of all of the solutes that you have in a fluid. So again, solutes can include glucose and amino acids and nucleotides and sodium ions and potassium ions and proteins. All of those are solutes. So you got lots of solutes, of course, out here in your extracellular fluid. You got lots of solutes in here in your intracellular fluid. And if you look at the total concentration of all of those different things, there's a term for that. It's called osmolarity. Ideally, your osmolarity is the same in your extracellular fluid as in your intracellular fluid. That's the ideal situation. You can have different subconcentrations of substances inside and out, and I shouldn't say can, you do, in a healthy cells have different solutes concentrated on the inside versus what's concentrated on the outside. But ideally, the osmolarity, when you think about all of those solutes together, will be the same. The overall concentration outside versus inside will be the same. All right, so let's take a look at how that works. So we're going to look at it from kind of a laboratory experiment perspective here. Let's say in a laboratory you had a tube, a water-filled tube here, and you've got a little membrane down here at the bottom of the U. And let's say over here on the left-hand side, you put in a solution with a low osmolarity, low concentration of solutes. And over here, you put in a solution with a high concentration of solutes. Okay, the solute could be a sugar molecule like glucose. All right, so in this case, what we're seeing here, we're going to say that this membrane is permeable to both solutes and water. So it's a very permeable membrane. So water can move across, and so can the sugar in this case. So that's going to be different from a cell membrane. We've already said that cell membranes are not only allow sugars across. Sugars are hydrophilic, so those are only going to move across if there's a carrier protein of some kind to move them across. Okay, but in this case, everything can move across that membrane that we've got there in the fluid, and diffusion is going to take place. All those molecules are in random motion, and over time, as they sit there, you're going to have water molecules moving this way, you're going to have water molecules diffusing that way, you're going to have solutes diffusing that way, and you're going to have solutes diffusing that way. So over time, everything's going to even out due to the random motion of diffusion. All right, but let's look at a case more like what we have with our cells, where we have a membrane down in here that is if we have a membrane down in here that is permeable to water, so water molecules can move across, but the solutes cannot. Impermeable to our solute, our sugar molecules that are there. All right, and the same thing, we put a solution with low solute concentration on this side, a solution with high solute concentration over on that side. What's going to happen? If we just let that sit, diffusion is going to take place. So your water molecules can randomly move back and forth across. The sugar molecules cannot. But as diffusion takes place, water is going to behave like a solute. It is going to want to move from where there's more water molecules around in a particular volume to where there are fewer. Diffusion is going to happen in that direction. You're going to try to equilibrate or even out the amount of water and the concentration of solutes on each side of that membrane. So over time, what you would see if you actually did this experiment in a lab is you would have a net flow of water in that direction. That side would take up water until you had an equal concentration of solutes on this side and this side. So you would actually see the elevation of the fluid on that right-hand side of the U-shaped tube there go up. And you could do this in a laboratory. You could use something like dialysis tubing, which is used for dialysis patients, to mimic that type of process. All right, and if you notice there, that continues until you have your solute concentration even out on each side of that membrane in the U-shaped tube. Okay, and so that is that's how your cell membranes behave. If you have a cell with a plasma membrane around it, you've got water inside and outside, you've got solutes inside and outside. And as we've already discussed, most of the solutes in your intracellular and extracellular fluids do not freely move across cell membranes. Most of them are blocked from doing that unless you got some proteins that are going to allow them to move across. Water molecules, though, are capable of moving in and out, and that's osmosis. Okay, so there goes my dog. Depending on the concentration of your solutes, just like we saw there in the U-shaped tube with a little lab experiment, the osmolarity, or the concentration of your solutes you have out here versus in here, will influence the net movement of water. Do you have more water that flows out, or do you have more water that flows in? It also influences the movement of water between different compartments in the body, such as from the blood to your serous fluids in the peritoneal cavity, or from the blood and into your tissue fluids that surround your cells. And yeah, we'll learn more about that later. Okay, so so we said the solute concentrations inside and outside of a cell are going to influence how much water moves in or moves out. And so there are some new terms here, and these are very important terms for you to learn. They're called tonicity terms. And tonicity, but the definition for that is the ability of a solution to cause a cell to shrink by losing water or to swell by taking up water. So if you have a cell and it is sitting in a solution in which the solutes are equally concentrated inside and outside of the cell, that solution is said to be isotonic. Iso means same. Now you'll see that when you're working with patients, because you might hook up a patient to an IV bag and give them isotonic saline solution. So that is water that has salt in it, sodium chloride, that has a concentration, the salt concentration is equalized with the expected solute concentration inside the patient's cells. And again, ideally you want your body fluids to be isotonic to the fluids inside your cells. A hypertonic solution is one in which the solute concentration, hyper means elevated or high, you have a greater solute concentration outside the cell than inside. A hypotonic solution, you have a lower solute concentration outside versus inside. Okay, so how in the world does this influence whether or not a cell takes up water or loses water? All right, so over here on the left-hand side, so this is these are actual pictures of red blood cells. They really do look like little red candies. And this is a happy red blood cell because it is surrounded by an isotonic solution. That means that the fluid that it is bathed in has the same concentration of solutes out here as inside. So again, those solutes can be different, but if you look at the overall concentration of all of the solutes together, if it's the same inside and outside of the cell, that cell is said to be in an isotonic solution. I notice little arrows here. They're showing you water is still moving back and forth across the cell membrane because it can do that by osmosis, but the same amount of water moves out as moves back in. Everything is already in balance, so you don't need more water on one side of the membrane or the other to try to equalize the solute concentrations like we saw with the U-shaped tube in our little lab experiment. Okay, now what happens if you have a cell sitting in a hypertonic solution? So if we got a little cell here, and let's say the little X's are solutes, and you've got more solutes out here, more concentrated outside, fewer on the inside. What's going to happen with your water in that situation? Again, diffusion's going to happen until you equalize the osmolarity or solute concentration on each side of the membrane. So in order for that to happen, you're going to have more water flowing out than is coming back in to try to equalize the concentration on each side of the membrane. Okay, so when that happens, what's going to happen to your cell? Your cell is going to shrivel because it's losing water. All right, now what about the opposite situation? What about our opposite situation over here? Let's say you got a cell and it has a greater concentration of solutes, things like electrolytes and glucose and amino acids and proteins. Let's say the solute concentration is greater on the inside of the cell. What is going to happen to the water? The water is going to diffuse by osmosis across the cell membrane to try to balance out those solute concentrations on each side. So in that case, water is going to flow inside the cell, and then this is going to be what happens. The cell is going to take up water and it's going to swell. And if a cell takes up too much water, it may burst, or lyse is the term for that when that happens. So if you took some of your red blood cells, for example, and put them in water, pure water, which has no solutes in it, those cells would take up, because they're sitting in a hypotonic fluid, lower solute concentration, they would take up lots and lots of water, and they would swell, and they would eventually burst. All right, so what's the easy way to remember this? And this is important to learn. Easy way to remember it is that water follows or is attracted to solutes. It's going to flow, more of it is going to flow across the cell membrane toward where you have a greater concentration of solutes. Or some people, just for simplicity, like to say that water follows salt, and the main reason for that is that sodium ions are the most abundant solutes in your extracellular fluids, and so sodium ions are actually the ones that really mainly dictate how much water is going to come out of your cells or enter your cells. And ideally, you want everything to be nice and balanced. You want to be surrounded by an isotonic solution, so it's very important for your body to maintain sodium homeostasis. You have to control the levels of sodium that you have in your body fluids very, very, very carefully. All right, so water follows salt. So water is going to move towards the saltier side of the cell membrane. So if the inside of the cell has a greater concentration of solutes, water is going to flow in, and the cell is going to swell. If the extracellular fluid has a greater concentration of salts and solutes, boom, the water is going to flow out when that happens. So I want you guys to start thinking now about, okay, well, what if I get dehydrated? You know, what if you got dehydrated on a hot day? You lost a lot of your body water. What would that do to your extracellular fluids, do you think, if you lost a lot of water? All right, hopefully you are thinking about how your extracellular fluids, if you lost water, you'd wind up in this situation. Your extracellular fluids would become hypertonic because you're not losing as many solutes as water. So as those solutes become more concentrated, that's going to suck water out of the cells of your body. That's going to be the response, and they're going to shrivel. And if it happens bad enough, your cells can stop functioning, and you can actually die if you become dehydrated enough. Or the reverse situation. Let's say you took in too much water. Water homeostasis is very important for your body as well. Let's say you took up way too much water without taking in proper amounts of solutes. All right, if you think about that situation, if you have excessive water and fewer solutes, you'll be in this scenario over here where your cells are bathed in a hypotonic solution. And in that scenario, your cells are going to swell. They're going to take up water. And, you know, that's not good either. They're not maintaining the proper internal environment they are supposed to maintain when that happens. So that's not good either. Either one of those types of situations can be bad news for a patient. All right. All right, so that's osmosis. Osmosis, again, as I mentioned, is a topic that will come up over and over and over again in your biology and health sciences courses, so you really want to start working on that now and understanding what we mean by hypotonic and hypertonic and isotonic and how that influences the amount of water that your cells are going to lose or gain. That's going to be a very critical topic for you. All right, I'm going to finally stop yakking, and then our last video lecture clip, stop applauding, stop cheering, but your very last one for chapter 3 and also for unit 1 will be about cell structure. So what are some of the functions of those internal parts of cells that we looked at a little bit on the first lecture clip for chapter 3.